Hi everyone, I'm Misha and I'm a member of 2025 iGEM team from Maastricht University, CoursePin. Within our project, we're introducing the iGEM interview series where we want to bring the state-of-the-art synthetic biology closer to you. In each episode, we sit down with leading scientists, engineers, entrepreneurs to explore their groundbreaking research, novel applications, and the thoughts that are shaping the synthetic biology of tomorrow. Please, let's dive in. Pranam, it's really a pleasure to welcome you as our first guest on our interview series. I'm Misha, MSP student from Maastricht University, focusing on synthetic biology.
And I'm Effie, a biomedical sciences student at Maastricht University.
So, before we dive into our academic and into your academic and industry journeys, could you maybe share a bit about your initial inspiration that drew you to the field of protein engineering, or maybe even biology in general?
Yeah, no, hi Misha, hi Effie, thanks for having me. So yeah, no, it's been a fun journey. I'm right now a professor of bioengineering and computer science here at the University of Pennsylvania, but to get here kind of took a lot of turns, which is not abnormal. So I actually started my science career kind of at Dartmouth College in the US, where I was an undergrad, but I studied religion, which is a kind of a weird starting point. Study a lot of East Asian religions and different faith practices. But I always knew I wanted to do science. My parents were scientists. They really appreciated teaching science as well. So my twin sister was at MIT, and I decided to say, hey, let's join her there. I transferred over, went from just taking religion and theology classes to just taking science, math, and computer science courses where I studied CS and molecular biology. But I think what really got me excited was this ability to really contribute to important problems and to solve them even while you were an undergrad, which you guys are doing now as a part of iGEM and other research projects. I got really hooked on it. I worked in a lab that was doing a lot of syn bio and obviously using algorithms to design molecules. And I was like, hey, this is like the future, even though at that point, a lot of the tools hadn't come out. So really got excited. I had done some research in antibody development at Pfizer over the summer to do for PD-1 targeting, which is a very large therapeutic area. But then, I was like, it made full sense to go to do a PhD. However, I didn't know really in what area I wanted to do it in. Was it going to be in computer science, which is what my academic training is in? Was it going to be in bioengineering? And so I couldn't decide. So I just said, let's go to the media lab at MIT. The media lab is a really weird place where you have people doing like robotics and software and I was one of the first people to do bio. It was just a real mix of random stuff. Did a lot of great work but my PI was actually the guy who invented the Kindle. And so we were like, okay, he's like, what do you want to do? And I was like, I don't know, I want to work on CRISPR because gene editing was just coming out. That was 2016, 2017 when I started my PhD, and it was really exciting. And so I was like, okay, fine. I want to do this because it's so going to have a lot of impact, right, in biology and engineering. And just very shortly, did a lot of algorithm development, made some new CRISPR enzymes with my collaborators, went to George Church's lab at Harvard, big synbio guy, converted kind of to engineering, bioengineering of ovarian cells, leading to the companies. We'll talk about that. And yeah, now the last thing I'll say is the lab is now working in the interface of developing algorithms to design molecules and doing a really cool bioengineering and therapeutic applications. So yeah, sorry for the long intro, but hopefully
No.
gives you a kind of a picture of the interesting journey that my research and I would say my academic interests took.
Yeah, of course, of course. And from the places that you mentioned, were
Yeah.
there any the people that really inspired you to become
Yeah.
who you are, and maybe some like the best piece of advice that they gave you?
Yeah, no, of course. I think so many steps along the way really influenced not only how I work academically, but also industry, right?
Yeah.
Also the companies that I've started. I would say there's two pieces of advice that I got that I think are useful to maybe share with you and with your audience. One, I think when I was an undergrad, my undergraduate research advisors were always very close to me. They would always, give me very good, kind of realistic goals. And the main point they told me is like, choose good questions, right? Like you can solve anything you want right now, especially as an undergrad. You can choose literally any direction you want to go in, major wise, classes wise, and research wise. So pick questions that you do not have to justify to people why they're important, right? Why did I work in like therapeutic design? Why did I work on AI? Why did I work in? It's not because I thought they were cool, but also because I know they would be useful. And if I told someone, hey, I'm going to use AI to design like therapeutics, people would be like, that's cool. Let me support you. So that was one thing. The other one is more company related. Both my advisors, Joe Jacobson at MIT, as well as my postdoc advisor, George, told me to do this. Never, when you create a technology, always patent it and never license it. I know it's very specific, but the point was always start a company off of anything you do. The worst that could happen is your company fails, but you're an academic. Your work grows with your research. But if your companies do well, that's amazing, right? And I want to support them. And obviously, there's a lot of payoff financially, but more importantly, you can get your technology to people. But if, at the end of the day, you have a lot of opportunities as a professor, as an academic, to start companies off of your research. And now, not all of them will work, but as you can see, like Gameto, my first one, and also the few others that I started have done pretty well. And it's given me a new way of translating my work.
So during your undergraduate years at Harvard Medical School, you dove into immunology, studying T cell regulation, and you even mapped how the PD-1 receptor alters T cell metabolism and function. Could you maybe share what it was like doing high-level immunology research so early in your career?
Yeah, no, exactly. And I was at MIT in my classes, and then HMS is where I did a lot of the research. And that was a really cool experience because, it's lucky, right? That the top research is just happening right like beside your dorm room, right? And so for me, that was the most important thing I wanted to do as a student, is just like, let me be a part of that. And I just wanted to join a lab that, first would take me as someone who had very little experience. I was a computer science student, right? So, like didn't fit with biology, but I knew this. I knew that research was going to have a big impact, right? If I was able to be a part of it, I would learn how to do high level research so that when I go in my future career, I would be able to translate that work. And the most fun part was we were not just working on some basic immunology, we were working on PD-1. And PD-1 at that time, that was when Keytruda, Opdivo were coming out. So everyone was curious, how do these antibodies and how does activating or inhibiting PD-1 affect T cells and the immune system. And we were able to show that by working on basic biology. Actually, by just doing that work, I actually got an internship at Pfizer during undergrad working on PD-1 agonist antibodies, not the blockers for cancer, but agonists for autoimmune diseases. Didn't work, whatever, things happen. But the fact was that helped me to see that basic science, right? Like understanding how PD-1 biology works translated to like therapeutic development could work so well. And all I knew was that the only thing I need to figure out is how do I inject computer science into that? And that's where the rest of my academic and research career took me.
And also, just like us,
Yeah,
you were a bachelor student at that time, as you said.
yeah, yeah.
But yeah, and you talked about the most rewarding part of this of doing this kind of research.
Yeah.
But what was the most difficult part for you?
Yeah, no, of course. I think the main thing was when you're an undergraduate, you really have a lot of, you actually are, I'm probably was the busiest Not, I mean, of course, at professor wise, I have a few companies, I have to teach, I have to do research, I have to do all grants, all this, but I actually think undergrads have it the most busy, simply because your priorities are not necessarily clear. Do you focus on classes? Do you get your A's, right, in your classes? Do you focus on research? Because that could actually give you a lot of great opportunities in the future. You have clubs, you have all these other things. I think the hardest thing is your time was limited and you still wanted to enjoy yourself as a undergraduate student. So I think, the difficult decision to just be like, let me just focus on research and, classes and grades will come, I will still go to class if I can, so I think you have to pick. And if you're going to do good research, I tell my undergrads in my lab now, you really need this to be your main thing, right? This can, like, if you want to do other things, great, but our lab may not be the right place for you. So I think that really prioritizing was the hardest thing. But I decided to prioritize research, and I think it was a good decision. My twin sister, who was also at MIT, she decided to spend pretty much every summer abroad doing really cool stuff. So I kind of was a little jealous because she got to go to like Italy, Germany, like Singapore, all these places, and I just sat in a lab for like 10 years, something straight or something. But I loved it. I wouldn't trade it for the world.
Yeah. Fair.
So afterwards at Duke University and now in UPenn, you started the Programmable Biology Group, which is integrating different algorithms to create programmable protein-based platforms for genome and cell engineering. Can you give maybe an example of a programmable protein platform your team worked on?
Yeah, absolutely. I think this idea of programmability is great. I know you guys do iGEM, you're in Duke's in bio. If you could just say, hey, here's a DNA sequence, I can edit it, right? That's what I did in my PhD, like making these CRISPR enzymes to edit any DNA sequence. What if you could, say, I want to make an ovarian cell, or I want to make a heart cell, anything, right? Programmably, like you tell me this, I get you this. We develop proteins during my postdoc, and now even in my lab here, that can say, here's the cell type you want. Here are the transcription factors, the molecules that you can use to make it, right? That's what the algorithm did. But I think what we mostly focus on, Effie, is this idea of most diseases are caused by bad proteins. Cancer, neurodegenerative disease like Huntington protein or P53 or something like that. We want a system to say, here's the protein, give me something that gets rid of it. That's it, right? Programmably, I can press a button and it'll give me something that can do that. And so what we've worked on over the past few years is getting really good at that problem, where we just published a paper today, like a few hours ago, it came out in Nature Biotech, where basically you give us a target protein sequence, no structure, because some proteins are not very stable, and we'll generate a short, like 10 to 15 amino acid peptide that binds to it. And not only binds to it, but remember how CRISPR works, right? Guide RNA and then a Cas9 that edits it, right? Guide RNA binds, Cas9 edits. So you're not just binding DNA, you're actually editing DNA. And so we said, let's do the same for proteins. You give me the protein sequence, we'll design a little short peptide, like a kind of like a guide peptide here. We attach it to an enzyme like an E3 ligase, a phosphatase, a kinase, whatever, and you're able to now bind and edit the protein. And so basically what we're doing is we're creating a synthetic system to edit proteins just like you would edit DNA with CRISPR. And I think that idea of programmability, if we can give it to people like you guys who are doing cool in bio, or to other researchers, like even companies, they're able to go out and say, oh, this protein's phosphorylated state causes cancer, STAT3. What if I could just like bind and get rid of that phosphate group? That would be, an incredible way of just quickly designing great medicines and therapeutic applications. And by the way, not just therapeutic applications, we just had work coming out on metal binding peptides for heavy metal sequestration. So really this idea of like binding and doing something is going to be a strategy for things beyond just medicine, bioremediation, food security, etc.
So yeah, the opportunities are endless, as you said.
Yeah, that's
So,
the idea.
but while the opportunities are endless, there like we know that there are a lot of like ethical regulations or many other big challenges
to getting this technology to the labs or to the market or wherever. So what do you think are the big challenges actually towards this path?
That's a great question. I think each technology carries its own risks, right? CRISPR, we know that. We've seen the ethical boundaries of editing embryos and using them in humans. And I think that has worked itself out relatively, where the focus usually starts with tractable problems, right? Basically, genetic diseases that we need to have cures for, and then we can move to more specialized applications. We have to create safeguards, right? Look, I'm an AI guy, right? That's my real academic work is AI. It is very possible, and I know now a lot of people are saying this, but I've said it for a while. It's very possible that someone could go into one day maybe ChatGPT or Claude, but now even some of the models that labs like ours develop, and if they're doing something wrong and they don't want to be ethically correct here, they could just design like the most pathogenic virus in the world and just go to IDT and make it. Like, it's easy to do, right? You could do that now. And I think that is a scary proposition that we're going to have to be careful of by seeing and evaluating what our models and our outputs experimentally are capable of doing. Think about it. For my company, Gameto, we were trying to make eggs from stem cells. Like, that was what I started the company to do, right? Me and Martin Varsovsky and Dina Rudenkovich, we were like, that was the first idea that underlied the company. Like, imagine if you just like made an egg genetically and then put it into humans. That would be you could make like synthetic humans essentially, right, from scratch. But I think what we decided to do was take more logical, more translatable approaches that were more ethically agreeable, where we are now just making ovarian support cells that will support eggs as they mature. And so I think that there are always ways to use good technology to do more tractable, feasible, and translatable problems first, and then say, okay, this is what we can do. What should we do next? And whether we can avoid those ethically ambiguous areas of research.
So now about your current research at the University of Pennsylvania.
Yeah.
It is advancing protein design using AI, as you mentioned, and language models. You basically treat protein sequences almost like a language, training models to design new peptides. So how do protein language models actually work?
Yeah, it's a great point. And I will say in the spectrum of protein design, which is obviously going to be huge for synthetic biology and has been, there's two ends. There's the people who believe that structure, right, three-dimensional structures are going to help us to design really good molecules. David Baker's lab, or like think of models like RF diffusion, Bindcraft, all the great models that do really well. I'm at the other end of the spectrum completely, where our lab dogmatically believes that sequence should be all you use, because here's my point. What if a protein doesn't have a good structure? Most disease-causing proteins don't. They just are spaghetti looking like things that you cannot solve via crystal or via AlphaFold. And so what language models do is very similar to what, if you think about ChatGPT or Claude or DeepSeek or these others, they basically said, here are a bunch of sequences, right? I'm going to hide pieces of those sequences, whether that's at the end or whether it's in the middle. And I'm just going to learn how to add those letters or uncover the hidden parts of the sequences or the sentences in that case. And by just doing that well and training a model, all the model has to do is do that. There's a neural network, a transformer that learns to unmask. By doing that well, we see first, we can learn grammar, syntax, semantics. We can learn how to make next tokens, next words. And we do that so well just by I'm not annotating the sentencing. This is a happy sentence. This is a sad sentence. This is the noun, the verb. No, I'm just giving it tags. And so in proteins, it's the same thing. You give it a lot of protein sequences. You just ask it to just uncover the regions of the sequences that I hid from it. And by doing that task well, just like in natural language, we learn not syntax, semantics, and grammar. You're learning structure, function, interaction, properties of proteins that are useful for engineering them. And what we do is we are a generative lab. We're not a predictive. We don't write like classifiers or predictors that much. We say, given the protein target, generate for me the peptide that binds to it, right? And that's what the paper today was one of the papers that we've shown that we can do that well. We've used those peptides just from like literally a language model. Those peptides came out. We degraded Huntington protein. We got rid of viral proteins for COVID pandemic. For pandemics, we bound to receptors. We did so many things just with a language model that models that like AlphaFold and RF diffusion, we did the experiments. They just didn't work on these really disease-causing targets. So hopefully I can convince you and your audience that sequence models are very powerful, and all you need is the sequence. You don't need to go and fold it. You get input the sequence and you can get what you want. One last thing I will say is that now at the lab at Penn, we're a lot more sophisticated in terms of how we're very theoretical almost as a lab. Like I have students who are like purely mathematical theorists, right? And people who are animal models. And we do everything in between. SynBio, high throughput screens, applied ML, everything in between. But what we're really focused on is this. This is the last thing I'll say. We're really focused on this question of multi-objective design with language models, meaning I don't just want a peptide or a protein that binds to my target. I don't just want an enzyme. I want a protein that is soluble, that has a high half-life, that is stable, that is not toxic. I want like multiple things. When I generate a sequence with a language model, I want all of those things to be true. And that, I think, has been an algorithm that has been very difficult, even in the AI community, to develop. We've started to do that. And we have peptides and molecules now that are like great across a lot of different things. And we now are testing these in animals as we speak.
Oh my God. That's an insane level of work. This is just mind blowing. How the AI model could do this kind of stuff.
Yeah, it's coming, right? Maybe one day Misha will be able to do just like ChatGPT by itself, like a natural language model could maybe do this. But I think natural language and like protein language are so different right now, and so that's why we're developing these more specific models here. But they work well, and we're like translating them to the clinic as we speak. So you'll see one maybe in humans in the next few years.
Nice. Also, going back to like you just gave a big talk about this CRISPR analogous system that targets the proteins. So maybe could you elaborate a bit more on how the system actually works compared to CRISPR?
Yeah, no, it's so I think I made the analogy, and I think it's a really cool like analogy because CRISPR is so easy. Hopefully you guys have maybe played around with it in the lab. Like if you'd have it, I could teach you in like 2 days. It's so straightforward. It's just like make your guide RNA, make your enzyme, complex them together, put them into a cell, the guide RNA binds to the DNA or hybridizes with the DNA, and the enzyme that is attached to the guide RNA cleaves and cuts the DNA and edits it. And so, what we do is we say, okay, you want to get rid of Huntington protein, which causes Huntington's disease. Okay, give me the target protein sequence, the Huntington protein. We'll design a peptide that binds to it from one of our models, right? Whether it's multi-objective or single objective, doesn't matter. Just make a peptide that binds to it. And then we will, in the lab, we will attach, like genetically, like plasmids, essentially, we will attach the peptide with a linker to the enzyme domain, the catalytic domain of an enzyme, like an E3 ubiquitin ligase, which adds ubiquitin and degrades a protein. We've had a paper on D-ubiquitinase to remove the protein. So you can take any enzyme that you want to do that does something to a protein. You can attach the catalytic domain to the peptide. Peptide will take it to the protein you want, and you'll be able to then modify post-translationally. And I will say this is something that needs to happen because in regular therapeutics, right, in pharma and biotech, you just want to inhibit the protein most of the time or to get rid of it. But what you're doing is you're just hitting it. But I think the future should be about modulating proteins, right? Most proteins are not caused by the presence of most diseases are not caused by the presence of a protein. It's caused by a protein getting modified and doing something bad. If we can reverse that modification, we can do it well. And we've shown results. If you see the paper today in Nature Biotech, you'll see results in various diseases, but also we're showing different post-translational modifications as well. We even have a language model that came out earlier this year in Nature Methods that was a PTM aware, post-translational modification aware protein language model. So now we're able to design binders that are very specific to PTM proteins.
Mm-hmm. Mm-hmm.
Yeah, so you have a lot of opportunity with this system.
Yeah, so we've seen how the anti-PD-1 therapies revolutionized cancer treatment by letting T cells function properly.
Yeah.
With your background in PD-1 biology and your current protein design work, there could be a lot of crossovers. So, for example, could generative protein design help create better immunotherapies, maybe synthetic proteins that modulate the immune system in new ways beyond traditional antibodies?
Yeah, that's a great question. I think that traditional antibodies are awesome, but like I just said, binding is just like one thing. And so I think there's so many cool immunotherapies that are coming. Obviously, antibodies I think are great. Peptides we're developing too, which are much better. Like antibodies, you have to get injection, right? You have to literally inject into your bloodstream or maybe do an intrathecal injection.
Yeah.
But peptides, I think, are we are working on orally bioavailable peptides, so you can just like take it by pill. So those things are going to happen. But I think there's another angle of immunotherapy that's happening. Think about CAR T, right? CAR T is where could these binders work very well in? What if you could design a binder to a receptor on, say, a cancer cell, and you can get T cells to be homing to that area, right? That's how CAR T works. At Penn, Penn invented here at Penn, CAR T was invented. And so we're really excited to use our peptides that we've designed from our language models. And what we're doing is we're working with collaborators and inserting those peptides into CAR T receptors, therefore allowing the CAR to go to different cells of interest and get rid of them. So I think that it's going to go way beyond just antibody development. And it's going to go more towards specific, personalized medicines where the AI is able to kind of manipulate and help the immune system target the pathogenic species that it wants to do.
Yeah, that's a very, very good point. That it moves to the personalized medicine more. So, but do you
personalized
think
medicine niche is very difficult, right? Because we also know, I mean, I have companies regulatory wise, even in Europe or in the US, to make a new therapy for every person requires a new clinical trial. And so I think that just as you're going to make personalized therapies, which we can do, we also want to create broadly useful therapies, meaning some people don't have the PD-L1 receptor. FA, so the PD-1 antibodies don't work for them. What can we create with AI, can we create a broader therapeutic modality that can work for everyone, regardless of what, if they have like a different genetic background or whatever. So I think that these are problems. I'm more interested in those types of problems where we're creating singular transformative medicines, right? Think about the GLP-1, right? The Ozempic or Wegovy. Like they'll pretty much is curing diabetes as we speak, but like that's a peptide. Did you know? It's just a little peptide, right? So if we can create the GLP-1, but for, cancer or for neurodegenerative disease or viral disease, that would be insane, right? That's obviously we've seen those work. You just take like a pill or an injection once a week. Like imagine if you had cancer and that's all you had to do, right? I think we're very close to that. And I think an AI is going to help us do that.
So yeah, and also now we want to go back to your industry part of your career. So in addition to your academic roles, you also co-founded the biotech startups like UbiquiTx and Gameto. So what motivated you to go to entrepreneurship alongside your research?
Yeah, it's an amazing question. And honestly, I'll tell you this, I didn't plan it. I'm not entrepreneurial. I will tell you that I'm very academic. a lot of venture capitalists ask me, Pranam, what would it take for you to leave academia and just like run one of your companies? Or I'm like, never. I'm like, I love being a professor, but you can't just have things operate in the academic lab. They will never see humans. They'll never see an application. So, when I was in grad school, I wanted to work in I wanted to do something cool, right? I was doing CRISPR, I wanted to do stem cells, and I wanted to make like these ovarian cell. Oh, I want to make oocytes because I'm like, that's the hardest thing to do. I didn't really think about why at that point. So, but I needed funding, and George was like, well, like you need to find some funding for it. And this is not a project where you go to like some government agency and they're like, I want to make an egg. They're like, they're probably like, no, like that's no, you're not going to do that. So, what happened was I was connected to a wonderful entrepreneur named Martin Zavarsovsky, flew me down to Philadelphia, actually, interestingly enough, where I'm now. And he said, okay, we can start this company to do this. But I'm like, no, 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 no. I don't want to start a company. I want to be a postdoc in Harvard and be a professor. And he's like, fine. So we went out and we raised money in Silicon Valley. We went out for like 3 weeks. We just like met people. I gave some talks on Zoom, and we just raised like $3 million, something like $2 to $3 million, and said, and he's like, okay, we have money. Let's go start a company. I was like, no, 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 no. I want to be an academic. And so what we did was we put that $2-3 million back into Harvard so that I could work on it as a postdoc, paid myself with that money, did the work, filed the patents, then said, okay, now because we basically said we started Gameto just as like a shell company of sorts. And we said, okay, let's put the patents at Harvard into Gameto. Our CEO Dina, who's amazing, she and I went out and raised $23 million to like, now the technology is mature from the academic lab. And now, just yesterday, Gameto announced a $44 million, basically like Series C, I guess, like Series B. We're in phase 3 clinical trials right now. Our first babies were born from the technology a few months ago. So who knew, right, that all I had at that point was like a little algorithm that could predict transcription factors. And now we have like full-fledged babies that are born as a result of the technology. I think that really, seeing that, now I can do UbiquiTx. We started off the peptide stuff that I was talking to you about. I'm co-founding another company called Atom Bioworks for mRNA optimization with generative models. So really, the point is this, it's that I just want to find the best way of getting the technology to be useful. Sometimes it's academia, sometimes it's industry. My hope is for diseases that are rare, where industry does not necessarily appreciate the need to like cure 5 children, right, for a disease. We can do that here in academia, add pen and translate it. But for technologies that have a broad utility for people all around the world, people who experience infertility, cancer, whatever, I think industry does a very good job of translating it, commercializing it, and getting it to people. So yes, I think that's overall the philosophy. And I'm glad that, I'm very lucky that Gameto, the first one, has worked so well, and, everyone now knows it, like, which is, some people forget I was a co-founder because I just kind of like did it, said, okay, I'm just going to be a professor now. And yeah, I actually have a meeting with them in like 15 minutes anyway. But the point is that it's great to be able to work with industry experts to get your technologies translated.
Yeah, that's an insane story. Maybe just a quick tip.
Yeah.
You mentioned like for the research you have to raise a lot of funds, of course.
Yeah.
So what's your pro tip?
Yes.
How
I think you have to be pretty diverse here, right? So there's different types of raising money, right? Obviously, in industry, you have to raise venture capital money or like, investor money. But that money is tied in the sense that they give you money so that you can do things, and then they take a piece of your work, right? They take ownership of your work. In academia, we pretty much, outside the university, we retain ownership. So in academia, the way you get money is primarily writing grants, right? Government grants. The check sizes, right, that you get are if you're really good, you get like $250,000 a year, which is like for us as academics, oh my gosh, so much money. For a company that's like nothing, you couldn't even survive on that money for like a month or something. But I think that the tip I would say is be diverse. Always look for places that are excited about your work. A lot of our money for our research in the lab comes from not only the government and in the US, but also private foundations. We work on rare diseases. Our stuff can work on like Huntington's disease, on Alexander disease, on these other rare diseases. And there are foundations that just want to have someone work on their and cure their disease, which I totally get. They're willing to fund your work because no one else will work on it. And so I think that being diverse in how you look for funding, whether it's industry funding, whether it's company, whether it's, just grant funding, will give you many opportunities. You just got to write. Like I literally spend most of my day just like asking, begging people to give money to my lab. And guess who does the actual work? Not me. I don't do research anymore. It's all my students. My hope is that I can give them the stability, the security, and the wonderful environment for them to do great work. And so that's my job, is just to be diverse in looking for funding.
Great.
So you're also a co-founder and advisor for Gameto, a
Yeah.
company focused on female reproductive longevity, reprogramming cells to fight infertility and one day even delay menopause, maybe. How did you start Gameto, and what is the company's ultimate vision?
Yeah, so I talked to you already about how I started, right? That whole process of raising money,
Yeah.
and just kind of started with an algorithm. But I talked to Dina a lot, and she's now the CEO and has done a beautiful. I mean, she's incredible. Has done a great job with the company. But I think we're right now focusing on the phase 3 clinical trials to be able to, help incorporate our ovarian technology, which basically matures oocytes that will never fertilize because they're bad eggs. You can take a bad egg, put it in these cells that we made, and it'll mature them. And I think the next steps is saying, okay, so we made these supporting tissue that can mature eggs. That's literally what the reproductive system does in females, right? Their goal is to create hormones and to mature eggs to hold ovarian reserve constant. And so I think the next step would be like, let's not just use this for IVF, for in vitro fertilization. Let's use this as an actual therapeutic, like cell therapy, right? Could we take these cells and literally inject them into patients and regenerate their ovary? That's what we're essentially making. And so I personally can't tell you much because I'm not on the ground at the company. Actually, the PhD student that worked with me when I was a postdoc, he became the CSO of the company, has done beautiful work there. They're working on building that platform such that they can transplant, hopefully one day, the cells that we're making into patients and be able to regenerate their ovaries. That would be the cure to menopause and also a lot of other reproductive disorders. So I'm super excited about what Gameto is going to do. It's growing at a fast pace. So if you're interested, anyone's interested in your, here, feel free to reach out and learn more.
That'd be wonderful. And yeah, and as you said, it's like these next steps, like building, maybe creating a cell therapy or something like this. It's very important. Like here we have an institute, Merlin Institute, and they're now trying to create all kinds of different. They're trying to create a wearable device which
Yeah.
has some cells
Oh, wow.
to battle the type 1 diabetes. So it's
Yeah. It's a thing. I mean, I think regenerative medicine cell therapies are so powerful for everything. I think the hardest thing is that these are very difficult to convince regulatory agencies to let people to put them into cells. If the cells go wrong, right? Say you put them into humans, like you could create cancers, right? You could create teratomas. And so I think that's why drug development and why we focus a lot on like these like GLP-1 like molecules, right? Because no one's arguing that's going to, like, if I put that into you and it works, people are like, that's a great drug. Like, of course, I'll just take it. But cell therapy, there's a lot of unknowns. So hopefully we can do well with these technologies, like your colleagues over in Europe, as well as here, to make these technologies safe, efficacious, and broadly applicable to different diseases.
Yeah, going back to Gameto, the research on women's reproductive health is often avoided because of the hormonal complexity, which creates then more challenges. But what inspired you to go into this research anyway?
Yeah, I think two things. I think largely dictated by curiosity, because I think because of scientifically, we don't know much, right? And that's a problem. It's because it's complex, one, but there's just not focus. Like, I think people don't see women's reproductive health or just women's health as a major area to do research in. So one was like there was a lot of untapped potential, and I wanted to solve hard problems, and this is a great place to do it in. Two, I think that I know, right? My mom went through assisted reproductive technologies, nothing like super difficult, but we were born as a result of that technology. And so I think overall it's helped so many people. And yet it's still kind of in vitro, reproductive technologies are still kind of archaic of sorts. There's not really a lot of advancements technologically. And so for us, it was like, well, this is an open area. People need it. It's enabled a lot of women to have children, to have, lead healthy lives. Like we should, and women are 50%, if not more, of this world. And so I think that it's something we cannot ignore, and it's an area that I want to continue working in. And whether new technologies can be applicable, we'll see.
Yeah, okay. And also on the other note, you as someone working in both the academia and the industry, like what do you think? Do you think being an active researcher makes you a better entrepreneur or vice versa?
Oh, 100%. I think
Yeah.
it's the first, 100%, because here's the thing. This is like more slightly almost egotistical, and I'm saying it from that angle, is that when you do well academically and your work is like broadly powerful, it gives you a lot of opportunities. People think entrepreneurship is just starting companies. Yeah, sure. People believe that, okay, Pranam must be pretty smart. Did all this. I'm like, I'm not smart, but like, thank you for believing that. And so they give you a lot of funding. And so you have more opportunities because you're visible and people know your work. So definitely it makes you a better entrepreneur logistically by doing that. And as a result of starting many companies, because you can only do that as an academic. If you're like a founder of a company, you can't just go out and found another one. Like if you're if you're a CEO, you cannot be CEO of two companies, but I can be a founder of many as an academic. But two, I think that as a scientist, right, as an academic, you still retain a lot of control over your work. You decide what you want to do every single day. If your company fails, which I hope they never do, but it doesn't affect you at all. You are still able to do really cool research and real good science. And so for me, that de-risking being in academia is super powerful. And regardless of if the companies work, we're going to continue doing great research. But I will say this, as an academic, you are pretty much perennially running a pre-seed startup company, right? A company that never grows because you're always like writing grants. You never have more than like a few 100,000 a year, right? Not like companies that have millions. So you learn, like entrepreneurship does help you to learn how to run academic labs too. So I think it works both ways. So yeah, who knows? We'll see if any of my companies work. Most of it is probably just, out of my control.
So looking ahead 5 to 10 years from now, the pace of biotech and AI is accelerating rapidly, definitely. Where do you think the field of protein engineering is heading?
Yeah, I think that's a very scary question of sorts, because I think a lot of these models, or even natural language models, I think GPT like models, of course they're going to be different, but I think that we're going to get so good that you type in something. Natural language wise, maybe I want a molecule that cures my disease that I currently have. Enter, you get the molecule either as a sequence, structure, whatever, and it works, like period, just straight out, no like testing many things, like one thing works, and then the path to regulatory approval is, as slow as it needs to be, but whatever, you'll get it through.
Mm.
And I think that's very possible. I think the other way is what I told you earlier, which is someone is going to take one of these models and design the most pathogenic virus in the world, and everyone will die. I think these are the two outcomes that we will either see, and I hope the second one is not true. But it could be, and it's very possible. The first one would be ideal, right? It would be such a nice way of just getting molecules, and not only therapeutics. I'm talking about, I want a protein or a peptide or a molecule that degrades pollutant chemicals, right? Or helps me to convert plants into nitrogen fixing plants, right? Like these types of, again, programmability, which is what we work on, will be so ubiquitous, right? Just possible everywhere, that we're going to be able to solve problems at a very, very fast. Do we need to solve these problems? For these problems, yes, we need to solve these problems. But there's a lot of problems we don't necessarily need to solve. We do not need to replace every human's job. Like, it's happening because economically it's viable, but we didn't have to do that, right?
Yeah.
Like, it's unfortunate, because I think AI will give us a lot of opportunity to do things that we need to do, but also we don't need to do, but we'll do them. And the world will look like a very different place. And I hope that place will be very positive, still very happy and productive.
Yeah, Pranam, I really feel bad about ending our conversation, because it was absolutely amazing. But we just have one final question for you.
Yeah.
So if you tomorrow woke up and all the tools you currently use, like CRISPR, AI, protein models, etc., they were all suddenly gone, but your knowledge and your creativity, you had your knowledge and creativity. Which project would be the first one you start from scratch?
That's a wonderful way of working on it. I think I would, knowing what I did, right, after doing it, I would probably put all, man, that's a hard question. I think we would probably try to get CRISPR working quick, because CRISPR is discoverable. Nature has made it. I think that you have to simultaneously really start pushing the models, because you have the ability to train these models. So everything you lost could be regained very quickly with AI, because it's just quicker and it gets you good things. But I think most importantly, I would, focus on, like, where were we making the biggest progress, and trying to pick up there where we were making progress towards cancer cures to neurodegenerative diseases. I think the main thing is these patients really need these tools, right? They need people to create solutions. And if we are able to do that rapidly, we're going to help them. So yes, I think it's a bad answer, but it is a mix of, I will spend 24 hours a day working on all of these things to try to get them back and working very well. So they're all great projects, and I hope we can continue working on them.
Nice. Yeah. So yeah, we would like to say thank you very much to you, Pranam, for being our first guest on our interview series, and for other MSPI Gem team members and Biomed Unleashed podcast team members for helping organizing this interview.
Thank you.
And yeah.
And thank you for having me. It was really great to meet you. Hopefully stay in touch. Hopefully if anyone listening wants to reach out, it's easy to find me, and we'll hopefully continue the conversation.
Absolutely.
Yeah, thank you very much.
Awesome. Thanks, guys.
We might even have some questions about our protein design for the.
Yeah, come to me. Use our tools. That's the hope, right?
Thanks for listening to the new episode of our interview series. If you enjoyed it, don't forget to like, subscribe, and share it with your friends. See you in the next episode.